Thymus. Thymus is a multilobulated structure because the capsule of thymus divides it into lobule by sending incomplete septa called trabeculae. Each lobule has a darkly staining outer cortex and lightly staining inner medulla. Lymphocytes in the thymus are called as thymocytes. Cortical thymocytes are immature. Medullary thymocytes are being mature. Thymus is a lymphoid organ, but it does not have type 3 collagen fibers. Therefore, in thymus, we have thymic reticuloepithelial cells. Thymic reticuloepithelial cells are slightly larger than lymphocytes and are more pinkish than lymphocytes. Thymic reticuloendothelial cells have multiple processes that keep interconnecting with each other so as to make a reticulum in which the lymphocytes can be suspended. Thymic reticuloendothelial cells protect the lymphocytes from the antigens in the blood, and there is gradual exposure of lymphocytes to the antigens. Therefore, TREX help in making the thymic blood barrier and help in training the lymphocytes. When the lymphocytes are trained, these are called thymus trained tree lymphocytes. Thymic Hazel-Scope cells. TREX form concentric circles with degenerating center which is deeply pinkish. Multiple Hazel-Cope cells are seen in the medulla. They are a unique feature of thymus. Spleen. Spleen is an encapsulated structure and the capsule is formed by collagen fibers which also sends in some septa or trabeculae. There is no cortex or medulla in the spleen. The spleen has red pulp and white pulp. White pulp, collection of lymphocytes in lymphatic nodules, forms only 20% of the spleen. It has two components, splenic nodule and periarterial lymphatic sheath. Lymphoid follicles are filled with B lymphocytes which form the plasma cells and finally antibodies. Periarterial lymphatic sheath. It's a collection of T lymphocytes which are surrounding the central artery in the lymphoid follicle. Red pulp forms 80% of the spleen. Splenic sinusoids are seen. Red pulp has a lot of RBCs intermixed with some lymphocytes. Red pulp has two components, splenic sinusoids and reticular connective tissue. Reticular fibers surround the splenic sinusoids and this is called splenic cords of Billroth. Histology of respiratory system. Respiratory system has two parts, conducting zone from trachea to terminal bronchioles. Respiratory zone. It consists of respiratory bronchioles, alveolar ducts, alveolar sacs and alveoli. Respiration begins right from the level of respiratory bronchioles and continues beyond that. Trachea, bronchi and larger bronchioles. They have pseudostratified ciliated columnar epithelium with globular cells. They also have hyaline cartilage which provides rigidity and also keeps the lumen of tubes patent. They also have some smooth muscle which helps in bronchoconstriction or bronchodilation under the effect of autonomic nervous system. Changes in the epithelium as we move distally towards the alveolar layer. Height of the cell starts to reduce. Columnar cells become cuboidal, which finally transition into squamous cells at level of alveoli. Size of hyaline cartilage becomes smaller and lesser at level of bronchioles and finally lost. Globular cells are also lost. Cilia also reduce as we move from trachea towards the smaller bronchioles. Globular cells are replaced by club cells or Clara cells. They start to appear at the level of terminal bronchiole and respiratory bronchiole. 90% of alveolar surface is lined by type 1 pneumocytes. Only 5% of alveolar surface is lined by type 2 pneumocytes. Terminal bronchiole, ciliated columnar epithelium with Clara cells. Respiratory bronchioles, ciliated cuboidal epithelium with Clara cells.